So we've got a jam-packed schedule, so let's begin. First up, we will enjoy one of my favorite presentations each year, which is the Q&A with the Alliance's Scientific Advisory Council, the SAC. The Alliance's Scientific Advisory Council is made up of 10 researchers from around the world. They lead the Alliance when it comes to anything scientific and help the rest of us understand the scientific jargon. So please join me in welcoming representatives from the Advisory Council. That's Kuldip Dave, Gethin Thomas, Adriano Chio, Nick Cole, Gorrit-Jan Blonk, and our moderator for today, David Taylor.
All right, I think we're up. So thank you everybody for joining us today. We're very, very pleased to be here and very honored to be able to provide another Q&A for you this year at the Alliance meetings from the Scientific Advisory Council. Before we start, we're going to start right in the questions and answers. I wanted to quickly have a couple of quick acknowledgements. The first one is we're joined here, as you were informed earlier, by Nick Kuldip, Adriano, and Gethin. But we also have Dr. Caroline Ingra from Sweden, who is a member of our Scientific Advisory Council, Dr. Louis Barbado from Uruguay, physician scientist, Dr. Piera Passanelli from the United States, Dr. Liu King from China, and Dr. Janine Heckman, who we all heard last night give a wonderful talk from Africa, a physician there. So we are very honored to be here. And before we do the Q&A, I have one last piece of housekeeping to say, which is that we wanted to acknowledge well, you can be a Dr. Gorrit-Jan, Gorrit-Jan Blonk, who has been the board liaison for the Scientific Advisory Council from the very start and been the biggest champion to make sure that the members of the Alliance are able to receive information through the Scientific Advisory Council. And we are so grateful to have had you helping us along and for all the valuable conversations as you roll off the board. And we will not let you get very far. And look forward to many, many other great conversations moving forward. So thank you very much, Gorrit-Jan. You will be missed on the SAC for sure, and always welcome. I don't even know if I'm allowed to say that you're always welcome because we're a volunteer council, but you are always welcome everywhere you go. So, okay, we will continue on now with the Q&A. I am looking here, so I will be moderating. We also have so many great things that we can talk about. I am looking for questions and I don't see any. So you know what? I'm going to start off with something until I get a question. So please put your questions in the Q&A section of the program or of the platform, and we will get to those. But I think one of the things on a lot of people's minds is the tofersen trial. It's something that read out the top line results just about a month and a bit ago. So I was thinking that perhaps we might have just a little conversation on where the current thoughts are in the field around the tofersen trial. And I'm going to actually hand off to Dr. Chio, Adriano Chio, to give some thoughts on his read on where the field's at in terms of thinking about tofersen.
Oh, thank you, David. Well, that's a complex question and a complex answer. The phase 1 trial was very positive for a subgroup of patients that are the fast progressing patients. By the way, fast progressing patients are really a small group in the SOD1 group. Now we know that because it was very difficult to recruit these patients during the phase 3 trial. The results were not so clear as were those of the phase 1, but there are quite evident indication that the drug is working in some way, and all the endpoints were not significant, but were all on the same direction, which is in favor of the drug. And the drug was overall also quite safe. There were some very small problems. So I think that it's a good news for the ALS community, but it's not a totally good news because the endpoints were not met. Now you know that the Biogen decided to give the drug to the patients for free. We are starting, I think, everywhere in the world. I think that's a good occasion for the patient to receive a therapy that can do something for them.
Great. So thank you, Adriano. And so actually to follow up on that, we do have a few questions, but before we get to those, since we are talking tofersen at the moment, I wondered if someone on the council wanted to also talk briefly about Atlas and how that is now sort of the next step in looking at tofersen efficacy in ALS. Kuldip, you're unmuted. The others are not. So I think you've done this to yourself. Would you like to talk about Atlas, or would you like me to? Or
sure, I can add a couple of sentences. Good morning, everyone. The Atlas trial is a trial that Biogen is running in people that have SOD1 mutations but do not have symptoms yet. So what we call asymptomatic or pre-symptomatic carriers. And the idea is that if we can go even earlier, then the symptoms come up and we can reduce SOD1 levels, that we can even perhaps delay when symptoms come on, or perhaps even completely prevent symptoms from coming on. And so this is a brand new concept it's not a new concept in science. We have thought about prevention trials for a while. It's brand new for ALS, and it's really exciting that, you know, and as my colleague Adriano just said, perhaps the current results from tofersen's phase 3 study are in fact telling you that maybe telling us that you need to go earlier, and earlier the better. And so this is the hypothesis that needs to get tested, and it should be really interesting to see what happens.
great, Gethin, you would like to add?
Yeah, I mean, I think as Adriano was saying, there's now an extended access program for all SOD1 positive patients. So I think perhaps one of the problems with the original Valor trial was that, one, it was only 6 months long, and two, the patient numbers were small due to obviously low numbers of people carrying the mutation. But with the expanded access trial, it's going to greatly increase the number of people, and obviously for a longer duration now we'll be able to see the effect of tofersen. So it's almost like a two-pronged approach now with the Atlas sort of prevention trial, but we still have the extended access program, and hopefully that perhaps will show that one of the other problems was the patients just weren't receiving the drug for long enough to really get to hit those primary endpoints. So there's still a lot to be watching in the tofersen space.
Yeah, and I think there's a lot of data still from the original trials to be released, even long-term open label extension data. So that'll be interesting as well. So actually, before we leave tofersen, we do have a question in the chat about it to follow up, but also looking over to Dr. Nick Cole, who's a former fundamental researcher in ALS, and thinking about SOD1 as a target in ALS is another thing that I think gives confidence to some level that tofersen is, you know, something we want to keep exploring, because SOD1 is, you know, something we quite well know is a pretty toxic factor in terms of a lot of basic research backed up. You can just raise your thumbs up if you agree with me that we have a lot of preclinical research on SOD1 as a good target, or thumbs down if you disagree.
Absolutely, yes. I mean, it's the principle behind it makes very good sense. Preclinically, absolutely, yes. And you know, we have seen in the results from the trial that the actual level of the SOD1 protein does drop. So there is some indication of a biological signal there. So certainly, you know, if we can crack this pre-symptomatic concept and give the product at the right time, you know, we're hopeful. There are also other genetic therapies in the pipeline as well. So, you know, coming at it from a multiple approach, and it's a really it's a sensible, targeted way to do things. You know, we're all very much disappointed with the initial outcomes of the trial, but hopeful that, you know, there is something in there that will come out in the future. And we're having hopefully an update of some of the additional information at the symposium from Biogen. So we may get some more information on you know, what they put the information out as soon as they can, and hopefully there's more analysis, as you mentioned, that we'll get in the next couple of weeks at the beginning of December.
Sorry, David,
Yep.
one more thing.
Sure.
I think there's a couple of companies developing anti-SOD1 antibodies as well. Is that correct? Does anyone know how those trials are going or where they are?
So there's AP101 from ALS Pharma, and they're looking at both sporadic and familial ALS for SOD1, extracellular SOD1, I believe. And that's an antibody based. And then there's a few other ones. There's APIC Bio. It's got a AAV microRNA that's decreasing SOD1. That's not in trial yet, I believe. And I'm not sure. I'm trying to think if there's other specifically targeted. I know that there was a drive for a long time by Novartis, previously Vexus, to do an AAV9 knockdown as well. But I think that program was abandoned. Adriano knows, I think, because he's shaking his head.
Yeah, no, it was abandoned. I don't know why, but they decided that it was not a priority for the company. But I want to add something about the use of antibodies or therapies on SOD1. The basis of the therapy with oligonucleotides in SOD1 was the concept that SOD1 is a gain of function. So if you reduce the level of SOD1, you could have an effect, and we saw that there was an effect, so a positive effect. But there is another side that could be a presence of a loss of function, and we have some in the information that there is also a loss of function. So if you reduce too much, probably you have a risk of negative effect. So we are not really sure how to work on that, which is similar to what happens with C9orf. Really, we are really waiting for the results of the phase 1 because it's not clear if gain of function and loss of function are the balance between the two mechanisms.
For sure. And that's actually a great segue to C9, but we don't have a C9 question, so I won't do that right now. But Nick's was also a good segue for that. But before we leave tofersen, there was also a question about who qualifies for the ATLAS trial in the chat. And I believe it's fast progressors because they're really trying to get those with the specific fast progressing mutations to be able to and then all other ones in the countries that have had the Valor trial have access to tofersen through whatever special access or expanded access mechanisms exist in those countries. I think the other thing I would add to ATLAS, and I don't know if this is just something I was thinking, is for people who may be wondering, when you get randomized to a placebo upstream in Kuldip, you might have said this, that when people start to show clinical symptoms, which is sort of the endpoint for, so we call it phenoconversion, once they start showing symptoms, everybody would go open label onto tofersen, so that there's nobody who's sort of started symptoms of actually getting ALS and then still on placebo in that particular trial. So people may wonder that as well. So, okay, if there's nothing else to add there, we might switch gears a little bit. And actually, it's just a small segue because there was a question around whether or not the results from tofersen could be extrapolated to other ASO therapies under development, or whether every ASO should be conducted in a trial rigorously. So I assume that the question may mean, should we assume that the rest of them are going to turn out similar, or whether we need to analyze them to see what will happen, or whether we can think, oh my gosh, this is going to be the same with other ones, is that we're going to have to treat earlier. Does anyone want to speak to that?
May I do? Okay, now I think we need to look at each gene separately, because we cannot say that what happens in one gene is positive in the other. And the SMA history is quite clear. It was very effective, the therapy, much more than we expected originally, even if the mechanism is a little bit different in the SMA. So for each gene, we need to see. We have now fast trial is ongoing, and probably other will be. But as always, it happens in research. Every situation is different.
For sure. And I think, I mean, it's actually, if we were to talk about things that we're excited about, I think one of the things that I'm personally excited about moving forward is we're going to finally start to get some information about how C9orf72 modification in humans will be, because we have not had any readouts of that yet. And we have a number of trials that are ongoing to reduce levels of C9orf72, but that one has like a lot of interesting complexity as well in terms of loss of function, gain of function. So in terms of reducing it versus maintaining levels of the normal protein. So we have a lot left to learn, but it's exciting times. I think that's probably a really overarching concept here, is that we're learning so much from these, and tofersen as an example is something we learn a lot from the Valor trial and where we move forward. And anyone, just please chime in. You don't even have to mute, all of you, unless you're muting because you don't want me to ask you like I did with Kuldip. But please, this is like we're all sitting in front of everyone in the audience. Okay, so there was another question about biomarkers that came up yesterday, and obviously this is a word that is coming up everywhere. And so a non-scientific community would see that and say, okay, that's great. We need to do something about biomarkers. What are we doing? So does anyone want to give sort of an overview of, at least very quickly, of sort of where we're at in terms of biomarkers and where the field is thinking? I see Nick and Gethin have both unmuted. So either one of you, did you want to tackle that one, or do you want me to?
I mean, I would just summarize to say it's not absolutely perfect, and we're still really looking for biomarkers. We've got some great indications from neurofilament light chain. That's one of the proteins that really make neurons. So as they break down, you get these fragments. Imagine sort of scaffolding that holds neurons together, disintegrate, and you'll get pieces of scaffolding in the bloodstream or spinal fluid, and you can measure that as a measure really of the breakdown of motor neurons. So we really need good biomarkers. And going back to the point about gene therapies and all effective therapies, we need to get them into people living with MND as soon as possible and as early as possible. You know, there's the sort of analogy that you get in these treatments which may be effective. You can't give them to people until it's really too late, and there's so much breakdown of the system. So we really need biomarkers for more rapid diagnosis, so that we can give things to people earlier in clinical trials. And there's many different arguments for that. So neurofilament light chain is one of them. There's a huge amount of work pushing towards finding biomarkers, which is effectively seeing if there are these things present in people living with motor neuron disease versus people without in control. So there are chitinases, neurofilament light chain, neurofilament heavy chain. Neurofilament light chain is really the lead one at the moment, but just to mention again, the symposium, which is in December, we've got a huge number of posters and presentations on different biomarkers that are being looked into to really try and pin that down. So, you know, we're not there yet, but the amount of work that's going on is more than there has ever been before. So I think, you know, we'll find a combination of various different ones, which are going to help. I don't know if you want to shed any more light onto that, Gethin.
Yeah, I mean, as well as the sort of biochemical or blood biomarkers, we've also got a lot of work going on the what you might call imaging or neurophysiology biomarkers, to try and look at the disease progression in a slightly different way, and actually looking at what's happened to your nerves, almost look at the actual nerves themselves, the neurons. But I think the challenges there are making it reproducible and making it easily sort of translatable to many different labs around the world. It's quite often need specialist equipment or very specialist skill sets to do it well. But I think the next step of this approach is to make it more available and more applicable, and to sort of homogenize the approach a little more. So There may be a two-pronged approach to this, as well as your blood markers you may well have using imaging, etc. as well to give us different sort of viewpoints onto how the disease is progressing.
There's also the what a potential urine marker as well that you guys are funded in Australia with Mary Louise Rogers.
That's right, P75, yes. So that does look pretty interesting. So that's, I think, being incorporated into a number of clinical trials at the moment. And I think I would assume, I don't actually know, but Mary Louise is probably presenting at the symposium somewhere about that data.
Yeah.
And David, one quick thing to add, another really interesting biomarker would be to measure TDP-43 itself. This is something that has been done in other diseases where, they, for Alzheimer's measuring beta amyloid, or for Parkinson's measuring alpha-synuclein. And the idea that this is the toxic protein in ALS, if there's a way to measure that in biofluids, that could also really change the way we look at this disease and the progression, and it may even perhaps be a diagnostic biomarker if we're able to measure it accurately in biofluids.
So I might actually add, based on a comment that Ralph from Biogen had put into the chat. So certainly there's a lot of great potential prognostic biomarkers to tell us how fast people are moving, or disease progression, perhaps neurofilament, or even diagnostic. But another one that I always think about when I think from a 50,000-foot view of where we've come in the field in the past 7 to 10 years or so, biomarkers that tell us that the therapeutic is doing what it's supposed to be doing, which is really great because it tells us one way or the other in the end, if we can get a good clinical readout, is that a good target or not? Did it actually do what it was supposed to do? And it always comes back to me, the Biogen trial for dexpramipexole, where they didn't get a good clinical readout, but they didn't know if it actually hit the target it was supposed to. And so Ralph had put into the chat about Neurone. And one of the things that Neurone did very well was it showed that it was intended to increase neurotrophic factors, and they seem to show that it increased the neurotrophic factors. It reduced an inflammatory marker, MCP1, and a couple others. So that is important, because as we do a readout with these trials, and it seems to be a lot of trials now are incorporating whether or not the therapeutic is doing what they expect it's going to do. And that's, I think, is a huge advancement in the field, because for a long time that wasn't the case. It was just clinical readouts and muscle strength and other things in SVC. So I don't know if anyone wants to add to that, or we can move on to the next. So biomarkers is a very, very urgent work in progress for the field, and a very active part of what we're doing, I would say. Okay, so here's a great question for Adriano. Based on the cytokinetics debate that you had with Amar a little while ago, is there a possibility we will eventually find out if ALS-MND has a 100% genetic cause? Now, I know you were on the other side of that argument, so maybe it's not fair to ask you that, but maybe you wanted to comment about how much.
Oh, sorry.
Oh, sorry. You might want to comment on how much of a percentage of genetics we think is connected to ALS, which is a tough question, but yeah, you're the person to answer.
No, this is a great question, I think. No, I don't think it's 100% genetic. And this is, there are a lot of information about that. For example, the twin studies demonstrated that probably only 60% of the disease is genetic. So it's the main part is genetic, but there is something related to other factors. Environment is one of these one. The research on genetics is going very, very well because of the characteristics of the genetic methodologies that are rapidly improving. The analysis of other factors is more difficult for several reasons, technical reasons. Environment is very complex compared to genetics. But anyway, the genetic component is probably about 2/3 of the cause of ALS, which is different from other neurodegenerative disorders. For example, in Parkinson's disease, it's much more low than ALS. And there are reasons about that. ALS is a rare disease. In general, rare diseases have more genetics than environment or other factors. Common diseases are more environmental than genetics, or genes are weaker. So it's I think it's the point for me. So I'm not completely on the side that of the debate that you could see in Perth.
Well, I think that's the point, right? They're supposed to be fun because you're taking a hard line on one side or the other, but you kind of believe it's a mix in a certain way. Well, you know, at some level, right? So I guess to follow up on that, maybe just briefly, because I know there's a ton of questions that come out about environmental factors, and would anyone like to comment on how difficult it is? And we're getting further on it, but it's really difficult to figure out the environmental piece, and the genetic piece is something much more tangible. Maybe I just said it there, and that's probably what. But does anyone want to add to that, or?
No, yeah. The point is that genetics is almost fixed. When you are born, that's genetics. Well, you can have changes, but these changes are much less important in neurons because they are perennial cells. They do not replicate. For environment is something that is changing during all the life, from the your in uterus life going to all the stages of the life. So it's very difficult to establish what you are exposed to during the life. You need to go back with your remembering, but it's very difficult because, for example, if I ask you what were your eating habits 10 years ago, probably you don't know that. You know something about that. So this is the reason. We are trying to overcome these problems using for example, environmental data from the environmental agencies. This is something that we can know quite well, but we cannot, the problem is that we cannot go back so much because this data, we have very good data in the last 20, 30 years. So maybe if a patient has 70 years, we can not really know what were the problems, the exposition when he was or she was very young. This is the problem with environment, but it's an area that is improving also.
Thank you very much for that. Does anyone want to add anything, or should we move on? I think we're down to 9 minutes already. It goes so fast. Nick.
Yeah, I was just going to add about the genetic components, that recent work showing that in people with no family history, some of these same candidate genes, SOD1 as we spoke about, C9, seem to have changes in people with no family history, which is a really, I mean, I don't know if you're going to come to this later on in terms of genetic testing and access to therapies, but it seems a very interesting that it's the same genes are involved in people that are changing in some change or causing M&D or associated with M&D, ALS in these particular people. I don't know if you want to touch on that at all, Adriano. Why is the same genetic components seem to be involved?
So Nick, maybe you want to clarify for Adriano. Is this for sporadic individuals, or?
Yeah. In terms of why we're now seeing data that shows that sporadic individuals have these same genetic changes as in familial cases.
The point is that now we know that sporadic is not sporadic. Is a mistake we did. Now we try to think about familial ALS or not clearly familial ALS. Because you can be sporadic for several reasons. One is a de novo mutation. We have a lot of patients with de novo mutation, so they are not present in the parents, they are present in the subject. Then we can have other causes of so-called sporadic. For example, very small families. In small families, it's quite difficult to have other members of the family with the disease, which is a problem, which is an aspect that is getting more frequent in Western countries, probably not in other countries. So the idea is that we need to abandon this term, and we have to understand is a mutated or not mutated patients according to the genes we have.
And actually, I think that's interesting too, as we get and I know that Adriano is a part of a lot of different committees to look into how genetic testing can be done across the world, but thinking about a large percentage of people that have been traditionally considered sporadic and how many more people, for example, with SOD1 may be in that group, even based on low percentages, than even familial, which is really makes you stop and think, if we do get therapies that are helping those individuals, how do we figure out we don't miss anybody? So it's so critical. Okay, so I'm actually I think we have 17 minutes in the end, I've been told, in the back end, which is good. I was originally thinking we were till 7:40. So I'm actually going to take a moment to address a question that we have, which came from Phil Green. And it's around why, it's a question that comes up quite frequently in the ALS community about when there's evidence of some people benefiting in a clinical trial versus everybody. And, you know, we've seen a lot of clinical trials where the initial primary endpoint shows that it doesn't benefit the wider group. And then when you look at a subpopulation, there seems to be some effect. And then when you also incorporate the fact that sometimes in trials, you will also have groups of people who will experience changes that are benefits for them while they're in the trials. So I think that that's a really important question for us to address. And I'm not sure if anyone would like to take that, if you would like me to start, if anyone wants to unmute, or Gethin.
Well, one thing I'd like to comment was that perhaps it's not always such a high bar for efficacy, but unfortunately our tools we have for measuring disease progression aren't great. So I think if we had better tools, then perhaps with such a heterogeneous disease, it's always going to be very difficult. Once we get better tools for measuring it, I don't think that bar will appear as high, because we will actually be able to show across patients with different progression rates of the disease that they are actually responding to treatment. So I think perhaps that is the challenge at the moment, that if we can get some of these biomarkers up and running and much more dependable and accurate, then I think that will help a lot of the challenges we have with getting good readouts from trials.
Adriano, I think you've unmuted.
Yeah, that's one of the great points that we are trying to understand better. It's related to heterogeneity of the disease. And at the moment, we have some difficulties in distinguishing the different patients according to different mechanisms, which is probably the reason why some drugs can be effective in some patients and not in others. The idea, the general idea is to use so-called posthoc analysis. You have all the database of the trial, and you go back to see if there are different group of patients in which in whom, the drug seems to be active. It was done in Adriano. The first trial was negative. They found a subgroup that was positive in terms of activity. And the good, they did another trial with this population, a so-called enriched trial, which is the best way to address the answer. The only weak point of the trial is that they did not put any biological biomarker. And this is probably one of the only negative aspects I can see. But it's sometimes difficult because now we are doing very small trials, so it's very difficult to find meaningful subgroups with small trials because you need relatively big numbers. But that's an area that we have to study and to work on.
And David, I was just going to add, you know, this is why the natural history studies are so important, because, you know, a study like Answer ALS, which can look at 1,000 or more patients and subtype them into, you know, this is a mitochondrial driven ALS, or this is inflammation driven ALS, or this is autophagy driven ALS. And if we can start to do that kind of subtyping, it will help both, you know, based on the mechanism of the drug, selecting the patient population that we want, in that trial. And as you know, to address what Phil Green is asking, not treating a general ALS population, but treating a very specific, personalized group that has that particular mechanism involved for their ALS. And so I do think that we can get there. For example, the ALS Association just recently funded Dr. Dave Ennist from Origin to look at 2 of the failed cytokinetics trials and look through the data to see if there were any subgroups where there were hints of efficacy, and if that kind of information can be used to predict responders in the futures.
Nick, you've unmuted.
Yeah, I was going to say that it's a great question, Phil, and it works both ways. You've got that post hoc analysis, an example being the Prelude trial, which is looking back at the data and seeing what looks like some response in people, and then discovering that, going back through the samples, that there's an UNC13A gene mutation. So then testing lithium chloride in that population of people. So that's a great approach, and as we've said, it's heterogeneous in different people in different ways. And so by coming together in these big collaborations as well, that's really important, because to get the numbers of people needed for those trials, for those subsets. So that's something that's happening now with these platform trials, with these big international collaborations. So, we're moving in the right direction, and it's definitely something that's being looked at and is really important for us to be doing. Thanks.
And Gethin, you've unmuted, so did you want
just to speak to Phil's second comment there about the sort of relative positivity around tofersen versus neurone. I think I don't know if there's a big difference, but I think it's people want to see the data. I think we're still waiting to see. I think, as far as I know, that the neurone data hasn't been published as yet. And I suspect if tofersen Biogen don't publish the tofersen data reasonably quickly, then there will be a degree of negativity around that as well. I think people want to be able to see the data to make up their own opinions to whether they can support the information coming out through press releases, etc. Until people see it with their own eyes, I think there's always going to be a degree of skepticism. I think when people can see the data, then we can all make our minds up and discuss it openly. Until then, we're kind of you're just kind of guessing to a certain extent, So I think that would really help people to be more open about to be more positive about neurone as well, if they can see the results and see the same things that Brainstorm see.
And actually, I would love to take that and pull that back to sort of an overarching question, because I'm curious about the four of you, your opinion regarding, the potential that if we do see or have some level of proof that there is a subset of people living with ALS, no matter how small, who we have proof that it benefits them, that there will be a drive towards figuring out how to get that for those people. Do you feel that that's fairly much the case, in the sense that when there's these subgroups, whether it is you're looking back to the Neuraltis trial with C-reactive protein, or whether you're looking at, Nick made a great point with lithium chloride, the trials around the world that were done, and then it looks like UNC13A, it may actually have some effect. If we can go ahead and get more rigorous proof that that subgroup is benefiting, we absolutely in general want to get that to people. Like, we want to find a way to get to that group. I think it comes back to the fact that we need to have that confidence that this is really benefiting that subgroup of people. Was that fair, or does anyone disagree with that? Or I think, I'll leave it.
I agree very much. I think ultimately ALS is going to be treated with a cocktail of probably drugs for everyone. It'll be personalized medicine. I mean, as Kuldip was saying, once we know exactly what mechanisms are disrupted in which patient, there will be specific drugs to treat those different mechanisms, and perhaps neurone and tofersen are just part of that that cocktail. for different patients. But we need to really have that data to be able to drill down into it, to be able to work out which drugs fit which patient.
Yeah, and thank you, Gethin. You made my point. I think the other point is that science has to drive the data. David, as you're saying, if there are subsets, tie a scientific mechanism to that subset, right? It worked in 5 people out of 100 because these 5 people had this gene or that biomarker or this, symptomatic profile, something that is scientific. It can't be, stories on the internet. And so I do think that, as I think Gethin said, scientific publications are going to increase confidence in a data set. And if for any drug, not just tofersen or neuron, any drug, if there is a subset of people where it works in, of course, we want to know how do you find that subset? Because if it benefits those 5 or 10 people, we should get them access to that drug. But those 5 or 10 people need to be tied scientifically to that response. And I think that that is what, frankly, companies have to do. They have, if a company runs a trial and finds that there are, subsets of people, let them know. First of all, were they on placebo or the drug in the first place, and let the community know what was the what the subset was on, and then secondly, what is it in those people? What marker, what gene, what profile that tells us that they would have a positive response.
Great, thank you for that. And I think that when it comes down to, as you said, stories, it's not necessarily that these are not these are real experiences that people are these are experiences people are having. And so it comes back to the sense that we need to have it's very difficult to understand what that means in the context of ALS based on the history of what we have in the disease with placebo-controlled trials and people on both arms who have experienced real things. And I think that it becomes a difficult thing, exactly as you said, for us to need clinical trials that can come forward and test those things so that we can have more confidence that going through all the procedures of getting this to the wider group or getting this to a subset of people is an effect of the actual therapeutic. And it's a hard question to answer because this is real experiences at the other end, but I think hopefully, we can convey that we feel extreme compassion for everybody and want to find ways to get real therapies to real, for everybody who can benefit, no matter what it is. And I think a great example of that would be thinking about the wider genetic testing and people that may be benefiting from a particular therapeutic that's targeted towards their gene, and they may not know that they have that genetic mutation. We want to find those people as a whole, and I'm looking at everyone to see if they agree with this, and that comes back to the sentiment that it's hard, but we need to find everybody who truly benefits from a therapeutic and get it to them. And I hope this has been conveyed by the group here, because I feel like we're all on the same page for that. Nick's unmuted. I don't know if Nick wants to add to that in the last couple minutes, or we can move to another question.
No, I'd absolutely agree. I think everybody from the M&D ALS community through to healthcare professionals, researchers, we all absolutely want the same thing, and we want to find effective therapies. Every person that's given their time and energy and sacrifice to going into a clinical trial, these are all really important case studies of information that we can learn from. So if we don't get that information, if we see, if there is something out there, or somebody's running around their garden after being in a trial, we want to know, are they on the compound or the placebo? Because if either way, that's a really important piece of information to build on for future trials. We don't We don't want to go forward with things that don't work. We want to go forward with things that do work. And if there's a subset of people in a trial that it seems to be effective in, then absolutely, all of us internationally, every organization would put all our weight behind trying to get that out there for people. There's no conspiracy going on here. We actually are all completely on the same page, and we're working tirelessly along with the community to try and find the answers to this. So yeah, let's be open and get all the information out so we can have a look at it.
I want to say thank you to Phil for that question, because that's the kind of questions that I think a lot of people have. And I appreciate the opportunity for us to be able to answer that. And thank you to the panel here to answering that question as well. And just to add that, you know, if there's follow-up questions to that by any members of the Alliance out there as we wrap up in the last minute here, please do reach out to us, to the SAC, and we will be happy to continue the conversation on that, because it is a nuanced and sometimes difficult conversation, and we want to have those conversations, and we want to, because there's, as Nick said, we're 100% behind trying to get the right information for you to better understand where we're all at in the field. So I'm at 7:49, and so I want to say thank you to Kuldip, Nick, Gethin, and Adriano for being here. And for those questions that were in the chat, we'll try to find a way to get you those answers that we didn't get to. And I'm so sorry I didn't get to them, but great, robust conversation. Thank you to the SAC. Thank you again to Gorrit-Jan. And I'm going to pass it off to Nick before I keep talking too long over my 7:50 deadline. Thank you, everyone.
Thank you all. It's difficult to know whether I'm in view or not. Hopefully I am, and you can see me. So thank you. What would we do without them? The SAC is such a brilliant group and adds such value to the ALS and MND community that, you know, we really appreciate you for coming together and forming the SAC. So thank you, David, for moderating. As always, just a brilliant moderator and tough act to follow. And thank you to Kuldip, Gethin, Adriano, Nick, and Gorrit-Jan for your time today, being with us. So what a wonderful and informative way to start today's presentation.